What I want to talk about today is a true example of how we go to the theme of this course, which is how we get to something that was once invisible to something that is now remarkably visible if you know the right techniques. And so the example of this image, which comes about, actually was developed for the Nobel Prize PR system. It's really a wonderful diagram that indicates on the left what we sort of knew of protein structure. Well, it's an interesting time to suggest 2013 is the point, but it's not a bad idea. But you've all seen this sort of rather lumpy potato-like model of proteins in a lot of textbooks. And what goes on in this diagram is the gradual evolution from something this lumpy to situations where we have increasing amounts of detail. So they become somewhat less lumpy and a little bit more filled in. And then you begin to see more detail as we go in through the different amino acid structures. And then eventually, as we get up to where we are now, people are actually starting to visualize what looks like individual atoms. And what I want to do is go through a little bit of the history of how this thing evolved. Because what's interesting, among other things, is that it changed technologies enormously. So it started with people interested in protein structure in the 1950s, 1960s. You're certainly in some way vaguely aware of this whole approach of X-ray diffraction, in which basically you set up a crystal of the protein that you're interested in. And by putting particular heavy metals at particular locations within the crystal, within the elements of the crystal, or by changing the angles at which you send the X-rays through, you end up generating these patterns of spots, which eventually can be converted into some sense of structure. And the classic examples, of course, were of Perutz and Kendrew, who did this marvelous analysis of the structure of myoglobin, thinking, of course, that they were going to eventually move to hemoglobin, but myoglobin was a simpler molecule to work with. And to show the change in structure of myoglobin when it absorbed oxygen. So it has a function similar to that of hemoglobin. The one you're probably much more aware of is the studies by Crick, Watson, and Wilkins, who received, that's the Nobel Prize trio, who received the Nobel Prize for their structure of DNA, which was done without Rosalind Franklin, although you probably are aware that it was Franklin's original X-ray images of DNA that prompted Watson and Crick to come up with their final model of what the DNA structure was like. So it's an interesting side issue. And then Franklin has been the subject of a number of these studies and reports because it's been sort of felt that as a woman, she was sort of treated differently from the others in the scientific community, from the men. And in a certain kind of way, cheated out of what should have been her Nobel Prize. On the other hand, it turns out, and this is particularly interesting, I think, that she became the director of the lab in which Aaron Klug worked. And according to his writings, it was her work that got him started on studying tobacco mosaic virus. And he did major work on the structure of tobacco mosaic virus, which is, you may remember, this is this virus that actually is a kind of cylindrical structure like this. And within the cylinder is the nucleic acid of that virus. Sorry, it looks a little bit like a worm, but that's the way it is. At any rate, because it was such a highly repetitive structure, he was able to use the techniques of X-ray diffraction initially to come up with the model for what the structure of this thing was. But X-ray diffraction clearly involved working pretty much with crystalline structures. And there was a great interest in seeing if you could get something finer with higher resolution. And the thought started to arise of using the electron microscope. This, by the way, is the picture of Aaron Klug. He received the Nobel Prize in 1962. One of the things that he showed in doing some of this work was that you could get a sense of the three-dimensional structure even when your images were flat by taking those pictures at different angles. And that's what these diagrams here represent. And he developed a mathematical approach, which involves Fourier transforms, which I'm not going to go into, in which you basically create a family of views of the object and then assemble them into a three-dimensional image. And I'll show you, he applied that image eventually to using the transmission microscope as well as X-rays. So here's the interesting issue that people came up with. When they started thinking about the transmission microscope, it was clear from almost the very beginning of the development of the microscope that if you used the electron microscope under what was then considered standard conditions to visualize something, your tissues basically disappeared at the moment the beam hit them, that they broke down, they were heated up, they were destroyed pretty much by themselves, okay? Because of what he's describing here. This is from, again, Klug's Nobel Prize lecture. He says there were preparation artifacts, that is, and radiation damage during observation. There was a problem in that the samples had to be evacuated, sitting in a high vacuum. And whatever water there was removed so that they were, in a certain sense, in completely artificial environments. And then there were lots of other issues with the physics of the electron microscope. So it took a while for both him and anyone else to really start using the electron microscope as a tool for high resolution structure of that sort. When he did, he actually used initially the equivalent of X-ray diffraction, which was electron diffraction through the samples that he was looking at. So if he had, again, this is tobacco mosaic virus, and this wonderful kind of cylindrical structure, right? And the idea is, because it was not only cylindrical, but highly symmetrical, you would actually get electrons diffracted through it. And so you would end up with some kind of electron diffraction pattern. And then you could extract that information into what eventually became this sort of three-dimensional model, which is still pretty coarse. I mean, there's no detail in the proteins themselves here, you see. And at the same time this was developing, a group involving, and one of these names you'll see again for the next Nobel Prize, Henderson and Unwin, put together a similar sort of model of the embedding of bacteriorhodopsin essentially in the membrane. I mean, they had sort of super thin arrays of the bacteriorhodopsin, which is known in a membrane to sit, if this is your membrane in the bacteria, the bacteriorhodopsin sits like this as a set of transmembrane helices, okay? And this is the image they came up with that sort of idea. So what happened was that basically Klug developed the mathematics for analysis of these kinds of images. Okay, and then 20 years later, actually more than 20 years later. This trio of people, this actually was a Nobel Prize that was awarded in 2018. I mean, it was quite recently, but their work came out in the late 1990s and into the 2000s. The three of them developed these modifications of the electron microscope to deal with just those sorts of issues that Klug had raised initially. And so let's see if I can give you this is a kind of an overwhelming diagram, but I think if I start with a diagram like this, and then I'll talk about the individual parts of the process as we go through it, then I'll show you some examples that you end up at the end, okay? So obviously, if you're going to study the structure of a protein, the first thing you need to do is make sure you have a pure preparation. So that goes almost without saying, but it's always worth saying. So you need pure material. But then one of the concerns that one has is that proteins actually live in an environment of water. And as soon as we put them into the either X-ray diffraction systems or conventional electron microscope, you end up removing that water. And so you're actually creating a completely artificial system. And so a great deal of effort went into this idea of what is called vitrification. And wasn't even clear when people started looking at it that it was possible to do this. The idea is that water, as it freezes, nucleates crystals. You get ice crystals formed as water freezes. And the question that Dubochet and others were interested in was, is it possible to get water to solidify without forming ice crystals? And it turned out that the way to do that was ultimately to use very, very small amounts of water and put it in something very, very cold. The initial idea was even to use some sort of antifreeze. So they mixed the water with glucose to sort of make it reduce the incidence of ice crystals. And then they basically, let me show you a diagram or an image, something like this, where there would be a sample basically on a solenoid, on some sort of magnetic switch that would thrust this probe down into a chilled chamber. And what was in the chamber was ethane, which you know of as a gas, of course, but it was chilled with liquid nitrogen to minus 196 centigrade. You may ask, why not just put it in liquid nitrogen itself? And it turns out that liquid nitrogen is not a very good freezing agent, because what happens with it is it boils off the moment it gets a little warm. That is, it's sitting in a temperature path or in a phase diagram. Here's sort of a phase diagram. You can imagine liquid nitrogen here, and gas here. And generally, this transition is very, very quick. So that as soon as something enters the liquid, it immediately, almost immediately turns into gas, and the gas ends up insulating what you're trying to freeze. So the way around that is to have ethane, which will not be at the edge of its liquid-gas interface, but will be seriously in the liquid when you add a sample to it. So the sample is frozen in liquefied ethane. And what the sample is an electron microscope grid, which you can sort of see in this diagram, with little holes in it, with little spaces. And the water that comes on there first is applied to the grid. This is a drop of water, sort of strange looking drop. And then you remove that drop, leaving just a thin film of water, and you plunge that into the ethane, and you end up with these little patches like this of frozen material. And that's what you end up looking at with the electron microscope. So that's part one. So now you've got it in the electron microscope. What do you have to do to make this thing work? Well, first of all, you've got to keep it cold. As it turns out, for many, many years, electron microscopes have been built with the possibility of a cold stage of a piece of metal that extended into where the samples were, that was cooled itself by running liquid nitrogen or various other things through it, so that you would have a cold environment within the TEM itself. And that was one of the criteria that one needed, because the idea was you would take this frozen sample and transfer it to the electron microscope, hopefully without it melting. And you'd have to have a little device to keep it cold all the way through, and then into the electron microscope where it would be kept cold. But then, remember I mentioned this problem that in sort of traditional electron microscopy, you would use an electron density, a beam of electrons, that was so strong that it would evaporate and destroy the material. So the solution for this was to use extremely weak illumination. Okay? Very weak illumination, which created its own problems. And the problems are sort of obvious when you think about them. If you're using very low illumination, then you have a real problem seeing any of your sample. First of all, because you don't have the detection capability for the weak electrons. And secondly, because the difference between the background and the sample is very little at that point. And so these things had to be overcome. And it turns out they're overcome by two advances. One advance was the development of extremely sensitive cameras. So they're called electron detector cameras, and they're sensitive enough to pick up this weak signal. That's only been developed for the last maybe 5 or 10 years. They're relatively recent, super detected, super sensitive cameras. But the other is it turns out to be something rather bizarre, that if you focus slightly off, under focus, you actually increase the contrast. Some of you may have had that experience if you're using even a regular light microscope, that what looks like it's in focus is slightly under focused, because when you focus, when you're a little bit under focus, your sample ends up with a slightly enhanced fringe around it. At any rate, Henderson and Joachim Franke, who worked on this thing, realized that it was possible to quantitate the amount of underfocus that was necessary. So you would generate these images and create them in such a way that you could see a little bit of contrast, but just barely. So here's an example. I'll give you a couple as we go along. Here's an example of what a field like that might look like. If you used conventional negative staining, which remember we've discussed a little bit as a technique for the TEM, you might be able to see something like these. But remember what these are is, here's your protein sample, let's say, and what is around the sample turns out to be this complicated array of negative stain. So the negative stain is actually all of this stuff here. And to a certain extent, it goes into your tissue, into your sample. But you've got a three-dimensional problem with it, as well as this question of how much penetration and how does the stain itself modify what you're seeing. In this cryo-EM image, that same protein is simply frozen. It's in a thin layer. And you can see, you can barely make out the structure. So by using this slight bit of underfocus, by using the more elaborate cameras, it's possible to collect these images, vague though they may be. And so each one is going to be, this sort of vague structure. But what you try to do with them is assemble them into a stack in which their similar images are combined. So the idea is this: if you have some kind, and I'll make it really simple for the moment, if you have a cylindrical sample, and let's say it even has a bar around it, right? And that thing lands on a flat surface. Well, one of the views of it clearly will be a view where it lands directly on the surface with its short end sticking into this thing, right? So you just have a view of a cylinder going straight down. You might equally well have a sample in which the unit is sitting on its side like this. And so you would see both of these as profiles of the structure. And as you see from these images right over here, it's possible to collect and reinforce all those images that, if I hunt around in here, look more or less like this, and all those images that look more or less like that, and stack them together so that you extract the essence of each one of those perspectives, of each one of those images. And that's what we're seeing in this set of 4 images here that represent different views of this particular molecule, which you really couldn't see individually by yourself. Okay. Well, if I'm going to sit here and do this, I'm doing it forever, picking out one molecule at a time, straining myself to see if I can actually see what's going on. And actually here I can see a pretty good example of one of those molecules sitting on its side. But most of them would really be a problem for me to go and sit and pick them out and then align them. So I would take all the ones that are on the side and kind of stack them up and then look at what's the common features in all of them. Well, this is where Joachim Franke and Henderson together wrote a series of computer programs, image analysis programs, that basically take these images, break them up into individual parts, automatically classify them, set them up into specific groups based on their shape, and then generate these kinds of images. So then you can take these kinds of images and start to work more to extract little bits of data from them. So let me go back to here, this is this section that I'm talking about of grouping the images by the angle of view. Okay, and then you create from that a 3D map using essentially those procedures that Klug had developed in the 1960s. That is the theory of tilting the sample, of assuming that when you have this set of images, it does represent in a certain way a family of molecules tipped in different directions. And so you can consider that if you now run your computer system to say, these are at different angles, let's now assume that they're all of the same structure and assemble this into a 3D molecule. So that was the basic idea of this process. And then you run into some other sort of unexpected problems. As you look at a sample, this is a very strange set of images, but as you look at this image, in the electron microscope, even though it's vitrified, even though it's cold, even though you're using a very low intensity of electrons to illuminate with, you get some almost thermal motion of these particles. And this is a diagram that says, look, if we take a movie of this image, that is, take a set of frames, okay, and then plot the position of these structures with respect to time, then what you find is that, in fact, they move around, okay? So now you could take a stack of this set of movies, the individual frames. So we have frame one, we then have the next one, and then we have the next one, okay? Which are different stages of time, that is, with slightly different movement in them. And again, realign them so that all of these are lined up in the same place, so that you would be able to say, well, what I really have is a set of identical images. Now that I have this set of identical images, I can look for the things that are common in it. Now, in this paper, they point out that you and I, and even they, would actually have a pretty difficult time seeing a difference between the figure in A and the figure in D, even though D is the corrected one. But they point out that if you do a Fourier transform, which is, again, something we're not going to deal with, except that the Fourier transform indicates a certain kind of regularity in the image. So what they're showing here is that in the uncorrected image, there's a fair amount of blur, while once it's been corrected for motion, this image is now much more easily correctable. I mean, you can see that there are lots of sections to it. Well, it turns out that the way the analysis is done of these images is you take this Fourier transform and you go through essentially the reverse of the math that generated the transform. But now what you've done is you've modified that transform to enhance certain features of it, so that what you end up with is an enhanced image of your sample. So what I thought I would do, just for the next couple of minutes, is show you some examples of what people have been doing now with this process, okay? So this is an example of the molecule that they're binding is streptavidin. And many of you who have done immunological work are aware of avidin biotin combinations. And so this is in A is this low magnification, low contrast image. And what you see in B is the accumulated collection of various views of that molecule. And then, although this is only, a small image of this material, the computer system is able to extract from these images then the essential elements that end up giving this kind of image of the protein itself. Okay? And so this is a stage in which we're now able to follow more or less the electron shells of the side branches of the amino acid chains. Here's another example from that same paper showing how they're really able to do this kind of very elaborate alignment, which then gets refined by slightly different perspectives and eventually end up creating this image at this point, which is the resultant of it. Okay. And then this is to take it even further, that what people are now able to do is develop the resolution. They're showing something about the progression of how much detail becomes possible over a 5-year period. They're really able to look at, see over here, some of the free loops within that amino acid chain. And here are the helices, very obvious. And these are coming not from theoretical considerations, not from the sorts of NMR calculations that you see in all of the standard molecular modeling. These are things that come out of electron microscope images with a sort of a remarkable amount of appropriate structure that comes from it. And let me give you two more examples. So what they're suggesting here is that you can take images like this. This is of a different protein. This is glutamate dehydrogenase. And they're showing you, again, something about what you can see. These are the images here, these processed images. And then the material that you see down at the bottom comes from certain regions of this. And these are regions, I think, that you can see over here. And they're actually looking at the level of where they're beginning to say, look, here's the shell of what we think is an amino acid, and that amino acid is tyrosine. And you'll notice they've drawn in a stick figure. But the resolution has been such that you can actually make out what you might call the electron shell of each of these side group amino acids. Let me now move over to this example, which is simply a gallery of all the different amino acids taken out of that particular set of electron microscope images. So it's really quite impressive as a result from a technique that I would guess, has really exploded within the past 5 years or so. It's really an extraordinary technique. It requires very specialized electron microscopes, very specialized computational facilities. One of the essays, one of the comments that I've read on it said that a standard lab to do this kind of work would probably cost $5 million. And so what's happening is that there are national laboratories that are available. If you have an appropriate sample and wish to develop a way of looking at your sample with these kinds of techniques, they have been funded by the government to support your work and actually to take your samples and look at them for you. So it's a kind of a remarkable example of a community coming together to share the value of really a pretty impressive technology. So I think that's as far as I'm going to take it today. One more example of something that was visible, that was not visible, that now is. In a way that I'd say it has the illusion of being truly visible, which is interesting. That is, it's not a theory, although it arises from theoretical processing. In some ways, the end results appear to be real. They appear to be something you could almost hold in your hand and see physically, rather than having to imagine it. So it's a very interesting movement in the direction of technology.